It's my distinct pleasure to introduce our speaker today, who hopefully looks familiar to all of you, Dr. Robert Lane. Bob began his career as an undergraduate at Johns Hopkins University, where he majored in Earth and Planetary Science and received his degree back in 1978. He then went on to do his PhD at Johns Hopkins, where he began his study on domestic and wild primates and cats. Then, when he went on to do his postdoc at the National Cancer Institute with Steve O'Brien, the genetics bug bit him, and he began his foray into technology and state-of-the-art techniques for studying evolution using some approaches that I'll talk about in just a second. Then after that, Bob became a research associate here at UCLA, after which he went on to the Zoological Society of London, where he was the head of conservation genetics for a number of years in the early 1990s. And then after that, he moved back to UCLA as an assistant professor and moved up through the ranks to where he is today. Throughout his career, Bob has received a number of prestigious awards, and I think one of the most significant ones that he received just this year, becoming an HHMI professor for both innovative research as well as innovative teaching strategies for teaching life sciences to undergraduates. What I think is quite remarkable about Bob and his research program is that he's interested in some big evolutionary functions that he's going to be telling us about, but over the last 30 years or so has been applying the latest techniques and developing the latest techniques. And to be able to do that and keep adapting to new techniques over a really long time period to powerfully address these big questions, I think is really impressive. And so Bob's going to tell us some more details about that today.
Thanks, Kirk. And I have to say what a delight it is to be giving a talk in front of so many familiar faces myself. Some of you I've known more than 30 years. That's really a wonderful thing for me. It's also a little frightening because any mistakes I make today probably will be remembered for another 30 years. So, but nonetheless, it's good to have friendly faces. Like Blair, I'm going to take a little bit of a retrospective approach. That is, I want to show you, beginning actually with my graduate work, that there really is a common thread to all the research that I've done for the last 30 years. So I'm going to travel back to when I was a younger person and begin to tell you actually how it came that I started working on dogs. I actually don't like dogs. I don't have one. They're sloppy and they shed all the time. So as an adult, I've really never owned a dog. But I like them for an entirely different reason that I want to tell you about. And that affinity for dogs, or working on dogs, led to a much larger question, that is trying to connect the diverse genotypes and phenotypes of dogs in a simple evolutionary model. And so I want to go over the data that we used to generate that model, talk about genes that are involved in body size, legs, and fur, and skulls, very briefly. And I'll talk about the overall question, which is very important to my future studies at that time, and that was the number of genes. How many genes absolutely influence traits in dogs? And then I'll put this all together in somewhat of a synthesis about what our model is for the evolving dog. So those are domestic dogs. Actually, my real desire was to try to explain evolution in nature. And so the second part of my talk will really be about that. And first defining actually what are the relevant population phenotypes or units in wild wolves, the ancestor of domestic dogs. And I call those ecotypes because they're actually related to ecology rather than to say distance. They really, wolves define different ecotypes. And given that, we can talk about local adaptation in these populations because they are adapted to different environments, and also look for deleterious variation where the populations are very small. So I'll run over that very quickly. Problem with all these studies is that you end up with a list of genes, candidate genes, that you think are under selection or deleterious. And it's very difficult really to publish these studies anymore because you need functional confirmation. Otherwise, it's a just so story. So I'm going to talk a little bit about functional confirmation and also just the frontiers of this functional confirmation in exotic species like gray wolves. And then briefly, another kind of functional confirmation. I'll mention something about comparative genomics, which identifies genes under selection, but at a much larger time scale. And lastly, in just the last few minutes, This is actually the places I've been, more or less, where I am now, and where I want to be in the future. So this is something about what my lab is sort of transitioning to do now, and that is reconstruct community ecology from samples of soil and sediment. So, why I actually do like dogs. When I started out in graduate school, I was really interested in adaptive radiation. I was an anatomist, much like Blair. This is where we met. And I was fascinated by rapid episodes of skeletal change and whether there are any rules to that change, whether there are any constraints. And it seems like dogs were kind of that morphologic radiation gone haywire, gone wild. And I wondered if we could look across dogs and find some rules that then would apply to wild canids. So I started out in morphology, measuring skulls and limbs, and my main notion was that see if there were regular changes in shape and size in dogs that I could model. And so much of this had to do with the 80s and Stephen Jay Gould and allometric growth and ontogeny and phylogeny. And these were my ideas were generated from. So I studied skulls and skeletons of a whole diversity of dogs. I actually went out and measured developing dogs, little puppies of Great Danes and pugs, small dogs, so I could see if there are any regular rules of change and growth that accounted for their morphology. And what I came up with, really briefly, is just the notion that it is kind of neoteny, at least in domestic dogs. That is, the growth of many dog breeds is prematurely stopped or slowed down. So what you end up with is the translation of a puppy-like morphology into adulthood. And I tried to really nail that down with actual growth studies. So this is in part what gives rise to the diversity of dogs, is that puppies start out looking so different from adults, and if you just kind of preserve the neonatal growth rates, you translate that morphology into adulthood, and in contrast, if you transition into the series of growth rates that goes into a normal adult, then you come up with something that's entirely different. And I put this together in a series of papers and models. One interesting outcome of this was the notion that some species, domesticated species, are predestined to be very variable because they start out in puppyhood looking very different with an abbreviated rostrum and a round, large, globular skull, and then they transition into the adult dog. And you can imagine if we stop growth or slow it down, then we can freeze these various morphologies and generate diversity. On the other hand, the difference between a kitten and an adult cat is absolutely isometric. That is, they're the same shape, although different in size. So if we stop growth anywhere along that trajectory, we don't generate diversity, at least in skull shape. And that was the basic notion that came to me from these studies about constraint and how dogs relate to, say, wild canines. It turns out if you looked at the diversity of dogs, they're far more diverse than the whole family Canidae of 36 species. In fact, some studies suggest their skull shape is more diverse than the entire order Carnivora. So this was a little satisfying, but a little unsatisfying, because we really didn't understand the genes that are involved in producing these phenotypes. And a little frustrating to me, because I wanted to be able to translate these results to understanding evolution in the wild. So fortunately for me, the dog was the 5th genome sequenced in 2005, and a group of us got together and said, well, we can look at that metrics to design a genotyping chip. Oh, this is the woman I went out with before Blair. So we knew something about the polymorphism in the dog genome and in a panel of dogs, and we were able to then take 160,000 polymorphisms SNPs and embed them, or get Affymetrix to embed them on a genotyping array. We immediately set out in genotyping a whole panel of dogs and wild canids, stuff that you could never do before. The decades of my life where we were just looking maybe at 12 markers in a population, suddenly settle organisms more. Sequencing a dog actually came from a white paper that a group of us wrote, arguing that dogs were a good model system, and that their phenotypes are partitioned in different breeds. So, for instance, chondrodysplasic dogs, the foreshortened limb dogs like dachshunds or corgis, there's 19 breeds that have that foreshortened limb conformation. And similarly, short-faced and large body size and small body size, there's all this phenotypic variation partitioned in different breeds. So we can do a case-controlled approach, essentially. We can take 19 breeds with short limbs and then normal-looking dogs, and we can ask what SNPs, what variants distinguish them. And that's kind of an association. This is basically, so a group of us got together, all our samples, analysis, Carlos Bustamante, was the leader in the analytical techniques. We tried to hire him at one time, even as a graduate student at Harvard. And then Elaine Ostrander, who had the dog sample, and myself working on wild Canadian dogs. The first genes that we looked at were for body size. This to us was the fundamental ecological parameter. I mean, after all, much of what is divided ecologically in nature has to do with body size. So there might be some profound genes in domestic dogs that also operates in nature and would set off our studies. So we took a bunch of small dogs, a bunch of large dogs, we did association studies, and immediately found that there was one variance near or in one gene, IGF-1, interleukin growth factor 1, that was associated with small body size in dogs. Now, with evolutionary theory, we can actually look for the signal of that kind of selection, and this is kind of a selective sweep analysis where what we've done is we've looked around this gene IGF-1, we've calculated the ratio of heterozygosity of large dogs here and small breeds here, and if there's a selective sweep for this gene in small dogs, there should be lower heterozygosity. And that's exactly what you see. That heterozygosity crashes right at the point of this gene. So we're using evolution to actually show that a gene is under selection. And of course, and also this FST is a measure of differentiation. The same thing too, it peaks right there. But that just means that small dogs and large dogs have different SNP alleles. Okay? And this is the actual tracing of heterozygosity in and about this gene. Red, small breeds, and then the blue, large breeds, and you can see that the heterozygosity of the variation is crashing in those small breeds, indicating a selective sweep. Okay. And here was, to me, a little bit of the sad story, that IGF-1, an incredibly potent factor in the evolution of small body size and breed, probably explains 50% of the genetic variation in body size. It is the master gene, probably the cornerstone for the evolution of small breeds thousands of years ago. So it explains a lot of the variance. In fact, just this and 6 or 7 other genes explain on the upwards of 90% of the variation in body size in breeds. And this is the beginning of the story when we began to look at other genes in dogs, like the one for foreshortened limbs. A single place in the genome was responsible for these foreshortened limbs. And it turns out this was a very unique phenomenon called a retrogene. It's actually a growth factor gene, FTF4, normally transcribed, and then reverse translated back into DNA and inserted back into the nucleus at a different, back into nuclear DNA at a different spot, back into the genome at a different spot. Those are called retrogenes. So the normal fate of retrogenes is that they're shut down, that they're not transcribed. In this case, this retrogene was actually expressed. So these foreshortened limb dogs actually have 2 active copies that are expressed. And this just proves this point here. These are foreshortened limb breeds like dachshunds. You can see they have 2 expressed copies. These are the normal juveniles with long legs, only 1 copy. And then these are adults where they don't have growing cartilage any longer and only 1 copy is expressed. So what happens is these 2 copies of this growth factor shut down the growth of cells at the epithelial plates and prematurely freeze those limbs. And so it is really true that the short-limbed dogs have a body which is inappropriate for a much larger dog. And this began to be the story of everything, actually. All the genes, coat color, coat texture, and this is another example here from coat color and texture. It's just 3 genes actually explain about 95% of the fur phenotypes in dogs. So this gave me the opportunity to actually say, well, I measured all those skulls when I was a PhD student. This is, of course, many years later. I still have them on a floppy disk. And we went back and we looked at those skull measurements and tried to see if they're associated with any SNPs, and they were. And this just shows you that there are high points of association, and it distills to maybe like 6 or 7 genes that seem to be explaining most of the skull variation in domestic dogs. And here's the little sad part. I wanted to see dogs as a model for natural variation. And as it turns out, things like IGF-1 for body size, the master gene, that's not really the case it is in the wild with normal phenotypic traits. And here, just showing you for, say, body height in humans, something on the order of 40 genes only explain a small fraction, 5% of the genetic variance in body size, whereas IGF-1 explains over 50%. So dogs are a little bit like hopeful monsters. That is, it's just a few genes. They're bizarre phenotypes. They would never survive in nature, and their genetic basis seems to be very unique relative to what we might experience in wild canids. For all these studies, we also looked at wild canids, and we didn't find these variants in wild canids. In terms of our idea about evolution and evolving the dog, of course, the dogs have a long history. Dogs and pigeons were one of Darwin's favorite species, and Darwin actually traveled around Britain, interviewed breeders, and asked them how they do what they do, and came up with the notion that it was mostly idea-driven selection. So if you wanted a dachshund with short legs, you actually selected individuals that had shorter and shorter legs. In fact, the dachshund, sometimes called the badger hounds, the notion was until recently they were selected progressively so they could fit down into badger holes and rout them out. Now we know it's just one retrogene that causes that phenotype in all 19 dog breeds. And so it isn't really this progressive model. We want to feel that we're smart and good breeders, and in many cases we are. But in the case of dogs, I guess they're not going to let me tell you about that. That's a bad sign. I don't like that. That's a little better. Guess I have to gather windows. I have no choice. Okay. All right. So, in dogs, it seems that rather than this progressive selection, it's driven mostly by the mutations. And once those mutations appear, like this retrogene, they're shopped around all over dogdom. And so they're put on the background of a terrier, of a mastiff, of a herding dog, and they appear very different, but in fact, they have a simple genetic basis. And we put this all together in kind of an evolutionary model. My student Bridget von Holt, who is here in 2010, graduated 2010, published this wonderful genetic clustering tree of domestic dogs. And there are a lot of notable aspects about this tree and surprising aspects. Of course, the first is that all the wolves do cluster together. So that makes sense. There's only one ancestry. You know, some thought, like Conrad and Lorenz, that actually dogs are so diverse they must have come from different wolf populations. That doesn't seem to be the case. There are also a number of ancient breeds, very ancient breeds, probably in the order, their lineages are in the order of 20,000 years divergent. And then there are the modern breeds, which are mostly the invention of the Victorian era, so the last few hundred years. The royalty like to have certain dogs. They're indicative of being very wealthy and powerful. And then, of course, if you make a dog like that, everyone wants the symbol of power and authority. And so they became very popular. But these breeds, the modern breeds, as we say, surprisingly group together with regard to function. And so we initially thought that ways to make a sighthound or a herding dog, so they would be all over the street. But actually, the sighthounds tended to group together, and these were names made up of breeders. You know, they don't have any idea about genealogy, but yet there was genealogical structure here. And so herding breeds grouped together, small terriers grouped together, mastiff-like dogs largely grouped together, and scent hounds, you know, I thought that would be everywhere in the tree. Spaniels grouped together, and exceptions were things like toy dogs. There, every breed we looked at was assigned to a different genealogical unit. So pugs, for instance, even though they grouped together in this cluster, were assigned to mastiff-like dogs. I mean, they derived from mastiff-like dogs. And so there seemed to be something going on there, and similarly, working dogs seem to have a diverse ancestry as well. Well, so to put this all together in a single model, what we think is that really there are two things going on. In sighthounds, the original sighthounds that were bred for sighthounds, maybe they performed better, and breeders would say, oh, I like the looks of that, and then they would cross and breed from that stock, and even though you had divergence, they came from a common ancestral stock. So that explains the similarity between function here and genealogy. With others, like the toy dogs, this is the shopping around of mutations. Dwarfism, once it appears in a lineage, breeders can cite that and say, I would like a dwarfed giant mastiff too. And we try to actually prove this in this paper, or at least gather evidence. And you can hear all the cases in red dots where we actually know the ancestry of these individual breeds, and the red dots accords with our notion of some mutation for small size or short-leggedness being shopped in to the breed. Like the pug here is a mix of a toy breed and a mastiff-like breed. So it fits our idea that humans are mostly taking these very discrete macromutations and moving them around all dogs. Okay, so that's the story of dogs as we see it. And there are various modifications of that story, but essentially dogs turn out to be a different paradigm than at least envisioned by Darwin. So here was my real interest. How about wild wolves? We've recognized that many of these genes we have identified in dogs are not what powers diversification of wolves. And here in wolves, at least we have a good case for them being diverse because we recognize today in wolves different ecotypes. There are ecotypes of wolves that live in the coastal rainforest off British Columbia. They mostly eat salmon and some black-tailed deer. They're small, phenotypically distinct. They're sometimes called the rain wolves. There are other wolves that we're more familiar with that are the Rocky Mountain wolves. They hunt much larger prey, like elk, moose, and deer, and live at higher altitudes. And there are wolves that live in the high Arctic tundra, even in the high Arctic islands. They're entirely different too. They feed mostly on caribou, but they also migrate over large areas. They participate in the caribou migration over 1,000 kilometers. I think it's one of the largest migratory pathways of any mammal now living. But they are migratory wolves. They don't breed with resident wolves in the boreal forest with which they overlap during part of the year because they're migrating at the time of reproduction. So these are very distinct entities. Early on, this is a study that another group did, not our group, but they came up with the idea that actually if we use a genetic analysis and ask how many genetic partitions are there within North American wolves, and that's what these points define actually, different genetic structure partitions. So these lines here are drawn around points that form a genetic cluster. But it happens that these points overlap distinctly different habitats. So here again is the notion of ecotypes reinforced by genetic differences amongst those ecotypes. And we took this quite a step further by using, say, remote sensing data, environmental data, environmental layers, and we actually showed that many of these ecotypes have distinct environmental niches or environmental packages. And we applied the SNP genotyping array. So instead of just 20 microsatellites in that first study, we looked at over 60,000 SNP markers. And what I want you to note about this is that we have recapitulated this pattern of ecotypes across the geographic range of North American wolves, and they correspond to distinct environments from boreal forest to Arctic and high Arctic, British Columbia, Atlantic forest, and those strange rainforest wolves right here that are actually the wolves that are invading, in part, Washington and Oregon. They're uniquely adapted to coastal environments. What I want you to note here is that these ecotypes actually, even though their average amount of divergence is fairly low, the mean is just a few percent, 4, 5, 6% divergence based on FST, that they actually have some genes or alleles which are actually fixed different from other ecotypes. So the maximum FST is approaching 1 in many cases. So there are a number of alleles which are unique or fixed different in these populations. And as you can imagine, those are candidates for selection. That is, maybe selection drove those variants to high frequency for reasons of adaptation. Maybe it was just random. We have to somehow look at the framework of what might be real in these populations. So what we did is we took our data and used a number of analyses, all with different assumptions, to try to look for these outlier alleles, outlier SNPs or markers that were far more divergent and had more homozygosity because of the selective sweep than be expected. This is work I did with John Novembre when he was here. And I have to say, this particular picture of him is like, says so much, because and it's not just the smug look on his face. It's actually this on his computer, because I think this picture was taken for Nature, is his famous similar system on wolves but for humans. It turns out the map of clusters in Europe kind of maps onto the PCA. So I think this is Iberia, and here are the British Isles, and Italy, and this particular figure has been shown, and I think Kirk will back me up on this, at more human genetic meetings than any other figure. It's right. It's like it has to appear 10 times during a meeting. But that's actually with humans. What we discovered is wolves are the same way. That is, if you use enough markers, you're able to differentiate wolf populations, which we'd spend 2 decades trying to do with just a handful of markers. This actually then is this joint statistic that John put together of homozygosity and divergence. So where a SNP is highly homozygous beyond expectations of random and divergent, and this is kind of a close-up of that peak, we can infer that SNP here, GDF5, that's the gene it's near, as under selection. And in fact, GDF5 is actually a remarkable good candidate, shows a very strong spline, and across all the other markers. So this is really reinforcing the notion that maybe under selection having something to do with annual mean temperature. Here's the other places we use. A very common one is Bayes scan. That assumes some kind of demographic model. It's the only problem. We don't know much about the demographic history. So here are all of the genes which are more differentiated, that is, markers near genes that are more differentiated than random, than predicted by our model. Here are some that are undifferentiated. This is kind of a blow-up. So these might be under balancing selection. These might be under divergent selection. Those are our candidates. And at the end of these studies, of which there are innumerable numbers in the pages of molecular ecology and other journals, we come up with a list. And this is where the fairy tale begins. Because first we look through this list and we say, okay, epiphysis, sorry. This is end of the bones, normally the liver, cardiovascular. So we start picking and choosing those genes which we think we can explain. That is, they make sense in the context of ecology. And we can also do geo-enrichment, geontology enrichment, and actually say, well, this whole class of pathway has more genes in it that are significantly differentiated. So that's mostly where those studies go. It's a little frustrating because it is just so storytelling, but it may be right. So what we did, that is what my student Raina Schweitzer did, and said, well, let's do one more test. These are SNPs that are near genes, generally not in genes. So they're SNPs that are tagging genes. Let's actually look at the genes themselves. Let's look at the exons, the introns, and then the promoter regions. And let's see if there are real differences in our candidate genes. So to do this, we designed or utilized this very wonderful tool called a capture array. And these are stripped out in beads. And what we do is we affix a nucleotide of a length, say 60 or 70 base pairs, that represents the core region of an exon. And then we add our DNA to these beads, and they bind, and we're able to actually use a magnet, literally a magnet, to pull all the beads aside. And then we can elute off or wash off the captured sequences from the DNA of a wolf that we've used. So each individual is captured in this way. And then that reduces our sequence effort many-fold. And so we captured over 1,000 candidate genes that we recognized as under selection from our previous study. And we also captured neutral regions that were far away from genes, so that's like our demographic control. And we did this in a wide sampling of wolves, most of which had been previously genotyped on that other kind, early SNP genotyping array that I mentioned. So we redid with these actual genes. We looked for non-synonymous changes, and we also redid all these tests, but this time on the non-synonymous or changes within genes. And we asked, are they confirmed? Do they have non-synonymous changes or changes in the promoting region? And are they still significant with this new round of tests, but this time in a different database of genes? And to our regret, only about 30% to 50% of the genes were confirmed. So our list of 1,000, maybe only 300 of them are real. But this is a much broader lesson for all the other studies that I've done. Some of these genes are interesting, and of course we can participate in storytelling more, but some of them relate to olfaction, pigmentation that we lost of geographic, and then lipid metabolism. So it's reasonable at least to discuss them. And then when we do a geoanalysis and ask, are any pathways, defense response to virus, are they correlated with our environmental layers? And in this case, they were with temperature. Makes sense. So the viral load changes as a function of temperature. And similarly, we can say other things, vegetation, yes, of course, different environments might select for different sensory nodes and also different aspects of anatomy. And we can do one more kind of more convincing test. At least we can look at where there's protein structures. In this case, LIPG, which is lipase, and so it's involved in lipid metabolism. And we can ask, is the change, the non-synonymous change we observe by this capture array resequencing, is that in an active site? So that's one other way of perhaps testing our candidate genes. Another interesting thing we can do is actually tally the genes that the alleles, which are unique and significantly differentiated in all our ecotypes. And when we look at that tally, it seems that actually the populations differ. Like the high Arctic has the most number of unique alleles that we find are significantly differentiated and may be under selection. Makes sense, it's a very extreme environment. But it also allows us a kind of ranking of these populations in terms of conservation importance. That is, if we lose the high Arctic and Arctic populations, we may lose a whole suite of genes. And it goes beyond what is now used with regard to just whether they're genetically differentiated or not. This actually focuses on the part of the genome that matters. What about deleterious variation? This is something that I've been very fortunate that Kirk is here. Here he is in his Halloween costume. This is actually, okay, this is at your Twitter. This is what's on your Twitter. So he's wise to be anonymous in this way, I think. And one of my students, Jacqueline Robinson, decided we wanted to look at deleterious variation because in very small populations, selection is very inefficient. So it's kind of like the flip side of natural selection in large populations, and as a consequence, deleterious genes will be expressed. So the loss of variation and the expression of these deleterious genes, especially if they're recessive, is of great concern. So I love this little fox. It's a distinct species, only found in our Channel Islands. It's about 2/3 the size of the Malin gray fox. Probably got to the northern Channel Islands about 8,000 years ago. May have been brought there by Native Americans. May, like the pygmy mammoths, have swum there. It clearly got to the southern islands, perhaps by Native American transport. If any of you have been to the Channel Islands, seen foxes, they're amazing, they're docile, they come up to you, We could catch them in little traps and just plop them in our laps and handle them, take blood and measurements in the teeth. If you do that to any other canine, even the tiniest of them, you're going to be in for a hurt. But not with these island foxes. They've lost their fear of humans. So this just shows their phylogeny. As expected, the northern islands versus the southern islands is based on complete genome sequence data. And here is the level of genome-wide heterozygosity. And the smallest heterozygosity in San Nicolas Island, where there may be effective size of 100, 200, it's an order of magnitude at least less than all the other species that at this time of the study the genome had been sequenced. Mosquitoes and the bottleneck dolphin. San Nicolas is way down there. So are many of the other islands. They managed to persist for probably 8,000 years with hardly any genetic variation. So what Jacqueline did with Kurt really was try to model this situation and use a demographic model, a very powerful demographic model. So here's a demographic model. San Nicolas, founded about 8,000 generations ago, went through multiple bottlenecks until the present day. And we can ask, does that model predict levels of genetic variation? Specifically, the type of genetic variation that matters. And in this case, this is a ratio of 0 to 4-fold heterozygosity. So 4-fold means that any change at that spot does not change the amino acid. 0-fold means that any change of nucleotide at that spot does change the amino acid. So this is the part that's under selection because it changes the amino acid. This is the part that mostly responds to drift. And this is the pattern we find amongst all the foxes. San Nicolas really high, so they have high heterozygosity and 0-fold sites, and then other island populations much lower. The gray behind this is actually what our demographic model predicts. And you can see our model predictions are such that they explain the pattern of loss or gain or retention of heterozygosity. So the selection is really weak in San Nicolas, as we predict, small effective size, and much greater in removing these probably negative amino acid changes in the gray fox. The other thing you can see here is actually the walk through the genome in 100 kb bits of the San Nicolas fox, which has the least variability. And you can see it's what we call genetic flatline. They're large parts of the genome, whole chromosomes, which have no genetic variation. This is the pattern in gray foxes. We don't see anything like that. So we call this genetic flatlining because how can a species persist for so long a period without any useful variation? There are some places, though, which have peaks of heterozygosity. Some 100 kb bits, which have higher heterozygosity. And we can ask, is this just a random thing? Or does this mean selection is saving some of the most important variants? So we did this too with a demographic model. And here are the results. The number of peaks that we observe in the gray fox and the San Nicolas fox are right in line with what we predict by our demographic scenario. So no, even though some of these peaks are like olfactory receptors, and our temptation would be to say, oh, well, they need smell, and that's important. Actually, it probably just reflects the abundance of olfactory receptors. There are over 1,000 of those in the genome. So this got us thinking, what's going on with the island fox? Maybe it is suffering from inbreeding depression, we just don't measure it. And they haven't really been observed to be inbred and any kind of fitness depression population. So Blair student Caitlin Brown actually took a look at skeletal indicators of inbreeding depression. And these are pathologic developments in the skeleton that reflect inbreeding. And they're fusions of vertebra or unusual abnormalities of the vertebra. And she tallied them from none to a pathology present, which has to do with developmental pathology before birth. And you can see that here's San Catalina and San Clemente, here's San Nicolas. So actually San Nicolas has fewer skeletons with developmental abnormalities than does the gray fox on the mainland. So there's no evidence there for inbreeding depression. Here's a case where there is clear inbreeding depression causing the extinction of the population. These are royale wolves. And you see this case is a huge amount of pathology. And in fact, in the last decade, all the wolves that have died had a pathology. So inbreeding drove that extinction. So this got us looking into yet another kind of simulation where Jacqueline with Kirk and his students looked at different demographic scenarios relative to the mainland and asked for strongly deleterious, moderately deleterious, weakly deleterious, and neutral variants. What would be the probability or likelihood that they'd be purged? And what you can see is strongly deleterious, that under a recessive scenario at least, that there's purging. That is, there are very low levels of allelic variation in deleterious genes. So here's the case where it's homozygous, and here's the total number. So the reduction all the way across the board in all these genetic scenarios, including the mainland, of a kind of weeding out of strongly deleterious variants. It's less so with moderately deleterious, and even less so with weakly deleterious. So these models support the notion that strongly deleterious variants, especially if they're recessive, that is, when they become homozygous, they're exposed to selection, they can be weeded out in these small populations, but they're still accumulating some sort of deleterious variation in more weakly selected scenarios. This was very important to us because we began to think about reintroduction strategies and how this relates to conservation. And so there's two different kinds of ways to be small with regard to population size. There's one way where the numbers are just reduced. So here are normal gray wolf populations. Here's a walk through the genome. This is the kind of picture of the runs of homozygosity, the distribution of certain sizes. And you can see in a normal population of outbreeding large population, there's high heterozygosity across the board. And a population which has a history of small population size, such as the Tibetan wolf, where that area of the Tibetan plateau has been reduced during the Pleistocene, so their population sizes were smaller, or the Ethiopian wolf that lives in the Ethiopian highlands, its actual area has also become reduced, but gradually. So these two populations, there's opportunity for purging. Small population size over a long period of time, selection can act to purge some of the load that is accumulated in large populations as recessive deleterious load. In contrast, these small populations on Isle Royale and the Mexican wolf that had gone to extinction in the wild, instead of this kind of distribution, they have large areas of the genome which are homozygous. So that part of the genome might be 10 megabases or whole chromosomes. They might contain hundreds of genes that are suddenly homozygous by inbreeding. And so any of the recessive deleterious variants in those large regions will be expressed. So we assert that this kind of situation is far worse than this kind of situation. And in fact, maybe as a source for Isle Royale, because they're bringing founders in from elsewhere, we might want to consider not using wolves from a large population because their load is high, because variants are occurring and aren't being purged out if they're recessive deleterious. Rather, use one that's kind of pre-adapted to being in a small population size where some sort of purging has occurred. They're pre-adapted to being at large population size. So it's shifting, I guess, the small population paradigm to some extent. I want to say that still, we're not quite satisfied with genes under selection or our deleterious candidate genes. And where we try to explore that more is in this case with black and gray wolves. Black wolves are frequent in North American populations. Generally, anything subarctic, they're about half the wolves are black. And this is due to actually a gene called the K-locus. It's a beta defensin. It actually functions in immunity, but for some reason, one variant, there's a 3 base pair inline deletion, binds with MC1R and causes the melanin depositional pathway to be activated. So it's a really real weird, freaky thing. But that is what this was described in dogs in 2007, and then we with Craig Barsh found it in wolves as well. And this is what we did actually. We had samples from Yellowstone population, black wolves, gray wolves, and we typed this variant in these black wolves, and it turns out all the black wolves here in blue had this particular variant, beta defensin, that we call the K-locus. The K-locus has remarkable properties that differentiate black and gray wolves. Turns out they have nothing to do with camouflage. In fact, wolves don't use camouflage to hunt prey. They actually pursue their prey over long distances. It turns out if you're homozygous for gray, and the black mutation is dominant, so you can only be gray if you have 2 copies of the wild type, it has higher annual reproductive success, whereas the heterozygote has higher annual survival, longest lifespan, long reproductive success. It's actually, there's a heterozygous advantage here. The matings tend to be disassortative, with black and gray mating more frequently than the other same color matings. And the homozygous black has the worst statistics. Generally, in 500 wolves, we only saw 4 or 5 homozygous blacks. So they have the lowest reproductive success. And this is work that Dan Staler did in my lab, both the ecological work and some of the genetic work with Dan Holt. So fitness differences between these 2 phenotypes suggest that it's more than just coat color, has something to do with immunity. And we explored that with a unique experimental design, beginning with Rachel Johnson, who convinced people who were killing wolves to actually send us some skin samples where this K locus is expressed. And so she established over 50 cell lines. And she also did CRISPR modification of one of these cell lines. So from a single homozygous gray wolf, she produced the heterozygote and the homozygous. And that was great because we had no homozygous cell lines. So to that, she added bacterial and viral antigens, even things that really appeared in nature, like distemper construct. And she looked at changes in gene expression, didn't find any by phenotype. And we began to think, well, it's wrong to really expect differences in expression. It's actually a function of the gene itself. Just the way it binds to the MC1R, it may bind to pathogens better. And so Tina Del Caprio in my lab has gone back to Duke, where actually Rachel Johnson is now. And she and Jenny Tong developed this new approach, much simpler, where we just grow up cell lines, like the CRISPR cell lines, and then we grow up bacteria, and we lyse the cells, and we mix them together, and then we subsample this mixture, plate it out, and see how many colonies are obtained. And this is a very striking result, I hope it's true, where the wild type, many more colonies develop when this mixture than in either the homozygote or the heterozygote mutant. Like 1/3 fewer. So this gene is providing some advantage with regard to this assay with staph. Another kind of frontier is single-cell genetics. And this is work done by Audra Hoffmeyer in my lab. I really love this work because there's a phenotype here. We can take sperm samples from inbred individuals and outbred individuals. And when you look at the sperm, especially of inbred individuals, they tend to be messed up. They have protoplasmic droplets or bent tails or coiled tails. They don't swim. In fact, in extreme inbreeding, more than 80 or 90% of the sperm are probably inviable. So here was our phenotype. And so we could pick like normal sperm, kink-tailed sperm, all these different phenotypes, and we could ask, how is their expression pattern different in single-cell genetics? In this case, we pulled 10 of each of these categories and are looking for expression differences between these phenotypes. And we're actually looking right on the mountain lines here in the Santa Monica Mountains. We're gathering sperm from those mountain lines. And this is just the relationship between heterozygosity and sperm viability. The other ways we can look at selection, of course, involve phylogeny and phylogenetic tree, and work that Annabelle Beichman is doing in my lab has to do with looking along the phylogenetic tree of otters, and she has a de novo sequence of the otter, and we also had one from the giant otter, and we're asking what genes are under positive selection. This is actual illustrations done by an undergraduate. There are undiscovered talents in undergraduates, and these are just some of her results. We find, as it might be expected, genes involved in fur morphology and the shape and weightiness of limbs, and also loss of olfactory genes because sea otters don't have. Similar work done by Daniel Chavez in my lab looks at the loss of, among other things, a toe, because in this highly cursorial African wild dog, brilliant animal, all dogs have a different coat color pattern, so we're trying to investigate all those things at the same time. Here are illustrations done by another undergraduate who actually took LS 110 and in her senior year, I think, decided I don't want to be a doctor or whatever, I want to be an artist, and took up a new profession. The last thing to has to do really with what I'm doing now. This fantastic effort that involves teaching 2 distinct to freshmen to try to get them to think about their careers and using environmental DNA, which is recovering DNA from soil and sediment samples, and trying to use that as a tool to teach them about the life sciences, all the career options they might have. So we have participated in a huge study across California, sampled 37 of the 39 UC reserves. We've recovered all kinds of species, from bacteria to mammals. We've engaged with 1,000 citizen scientists, like 350 undergraduates and counting, 18,000 samples. That's all sponsored by the HHMI. And these are some of our initial results. This just shows you kind of a beta diversity. The more hot the color is or cold it is, the more different that community is. Here's an example for plant composition. So these are generalized metabarcode primers that will amplify really all the bacteria and plant taxa there. So here are the plant orders, and these on the x-axis are all our different localities, and you can see there's ubiquitous taxa. There's taxa that only appear in a few localities, and in microbes the same kind of pattern. We can look at even individual networks of co-occurring at one place. This is work that's led by Rachel Meyer, who's the executive director of this whole project, and Emily Curd post. This is similar work done by Zach Gold, using a program Zach and Emily developed called Anacapa. And it's based on actually their first sampling off Anacapa Island. These are the species that are recovered from water samples that are then filtered, and fish and even birds are identified, their DNA from these water samples. We're pairing all this information with remote sensing data. We're working with JPL and NASA that have this wonderful new platform that they have up that's able to deduce 200 spectral layers. So environments can be dissected to the individual as to potentially what species they are. And so we're trying to provide them the species data, what's there, and then they're trying to backtrack from their spectral layers to how those species can be identified elsewhere. This is the huge team at Cal DNA. So there's I have two scientific postdocs that are really running these classes, that are doing a wonderful job, and then we're also paying part-time lecturer to help develop the curriculum, and I'm involved as well. And then the postdocs who are doing the science, and also my co-investigator at UC Santa Cruz, and then of course the wonderful staff at SEALS who are trying to make our class really. And then just want to acknowledge everything, and I have one more thing that come up here real quickly. So, happy Thanksgiving, everybody. Did I say Thanksgiving? Happy Halloween! Oh. Yes.
Oh, so I was interested in your the restoration, the part about the restoration work. That, so you showed these genetic patterns in the different populations, and you made the argument that you should not have the founder, say for IOL,
yes,
from well-mixed, like a population like Minnesota, which has a higher genetic load than the one from Ethiopia, which has a low genetic load. But one might argue that every introduction, what really matters is that the introduced population survives, increases when rare, and that's a primarily ecological phenomenon. And they have to survive in the rare. So you might argue if you had a found from Ethiopia, which is a completely different ecological realm, and you introduce something to IOL, which is in the same zone, that genetic variation is secondary because you just have to survive. So I was wondering, whether you had a comment about that, whether any new recommendations should be not taken into account, but in order for genetics and adaptation and all these things to matter, the population has to survive and increase transform numbers.
Absolutely, that's part of the argument, and I would take ecology over genetics in this case. But the problem is that when we inbreed, we don't have any choice in IOL because it's the only population that can hold 30 wolves. They're going to become inbred. So the deleterious recessives are going to be expressed. But absolutely, they have to come from a population that knows how to kill elk, because that's all that's on IOL. And elsewhere, there aren't the elk in that forested environment. So the actual survivorship has to come first. You're right about that. And this is something but it does question the small population paradigm, that the overall level of variation is always the principal thing you want to maximize, because a significant fraction of that genetic variation is deleterious variation. In fact, a large fraction of it is deleterious variation. So we're trying to turn that on its head a little bit and just get people thinking about what it means. That yes, yeah, yes.
Great talk, thank you. I'm curious about the coat color Caleb Benson story.
Mm-hmm.
Two things. One, I didn't fully understand the plot with the bacterial exposure and the lysed cells.
Mm-hmm.
Where the heterozygotes seem to be growing more bacteria.
They're going fewer.
I thought that, okay, did I just get that back?
They're fewer, yeah, I got it backwards. They're fewer colonies. So here's the wild type, more colonies.
Okay.
And here's the heterozygote and the homozygote, fewer colonies.
Got it.
About a third fewer colonies.
So the field related question is there any sign, like, so there's a distemper outbreak in Yellowstone wolves just a few years ago. Is there any sign of
Yes.
acting on these sites?
Yes. There's sign of selection that the black heterozygotes do better with a variety of challenges, from distemper to mange. So they do seem to be doing better. In fact, we put a model in place, myself and one of Kirk's students, and Jacqueline, to that shot I showed you of the genetic variation in wolves. But we put a model in place that seems to suggest there's active selection going on. This is Raina Schwartz's work too, in Yellowstone. That the only way you can explain the current ratio of black to gray, you have to invoke some kind of selection, even though they were only introduced in 1996.
You
Yes?
mentioned that the dogs have very few mutations that explain most of their variation. I was wondering if, that is that a particular sign of artificial selection on dogs? Are there other species that self-artificial selection have similar genetic makeups like that, or is that something particular to the species?
Well, I mean, it's particular to dogs, because unlike cattle or wheat, where specific phenotypes like less fat or higher growth rate were selected, in dogs, we're mostly selecting freaks, right? So individuals that had freakish morphology, because we wanted breeds that were very distinct and very different, that made them more popular. So it's a very weird kind of, we selected for novelty more than we selected for function in dogs. Yeah.
I was just going to ask, on the flip side of that, is there something about the weirdness of dogs, these large effect size genes, that make them in some way pre-adapted to domestication? Were they easier to selectively recolor to functions because maybe they're weird genetically going on?
Lots of people have suggested that, like the role of transposable elements and the rapid change under domestication, or some other genomic aspects that are unique to dogs, but it really hasn't borne up over the years. I think it's mostly that variants that would be quickly, like the short-leggedness, lost in the wild. In fact, there are records of achondroplasic foxes in the wild, but they don't do very well. They probably get eaten by other foxes. And so I really don't think there's that much unique with regard to mammals in the dog genome, except the fact that we selected freaks. Yeah, but we think that we've done a much better job, I think, often than we have. I mean, it's true some dogs are good scenters and good, at retrieving, and sight hounds and things like that. They have unique attributes, but we don't really, we didn't really study those behavioral attributes. Very difficult to quantify. There was just a study published in Nature, Ecology, and Evolution about a month and a half ago on these foxes that were domesticated, and it does seem like they have a cassette of genes having to do with domestication and behavior. Yes?
Yeah, so I want to go back to Priyanka's.
Oh, I should have picked Paul first.
Oh,
Yeah.
sorry.
No, go ahead.
I want to go back to Priyanka's question and the sort of assisted migration. What are the best populations to use? Because it's very generally important in conservation right now. And I guess the question is, if you look at several of those different populations that all have gradually reduced their load by selectively removing deleterious alleles, do you always remove the same ones in each population. Maybe you showed this, and
No, I didn't.
But do you always remove the same ones, or is that sort of removal itself sort of very site-specific, which would sort of argue that then you would only want to use that in a very similar or nearby population or something like that, as opposed to taking one from so if everybody reduces the same deleterious alleles out of the population, then maybe you'd sort of say, that's just a general phenomenon. They're just sort of better almost no matter what.
Each population has a different demographic history, and so each one is going to have a different history of the accumulation mutations. So, and the same with purging. Yeah, so
they purge different things.
They purge different things.
So then you really would say that purging itself is sort of site-specific.
Yeah, the specific amount of purging. The thing is, actually, you're establishing these very small populations. They're destined to be small for their entire history, and you're going to generate long tracts of homozygosity. That's just inevitable. And you're asking, do we want those tracts of homozygosity to contain fewer deleterious variants? Maybe so. And so that's what we're asking here. They have to be adapted to that environment. They have to be appropriate for that environment. But it challenges the notion that more genetic variation is always good, because much of that variation is deleterious and hidden in recessive deleterious variants. Sorry.
So I like your description of dogs as a selection for these traits for genetic freaks. And when you talk about the phenotypes, but I assume that there are also various SNPs that are associated with some compulsive behaviors that dogs have. So, herding is a compulsive behavior, pointing is a compulsive behavior. How many genes are typically involved in those sorts of compulsive behaviors? And given work that people are doing with human association studies and behavioral disorders in humans, are some of the mechanisms perhaps similar in terms of these compulsive behaviors?
Yeah, no, there's a rich literature on that topic. For the real compulsive, discrete behaviors, everything from dogs that run into like a shoebox when there's the lightning strike, or the mad cocker syndrome. There are all these disorders that dogs have for which the genetic basis, narcolepsy, several genes identified first in the dogs that are also found in humans. It's been argued, like in this white paper that we wrote to get the genome sequenced, which cost $30 million at that time, to get the genome sequenced, that dogs share more genetic disease with humans than any other model species. And they do. And they're understandable with regard to human behavior. So now there's a rich literature in this topic of very discrete disorders. Understanding what makes them so tame, what makes them so dumb or whatever, we don't know. And Bridget Van Holt has done some work in this field. It may have to do with the Barron syndrome, where there's a big deletion, the love gene, or they call it, where there's a loss of a large region, like spanning 30 genes. And the humans that have that don't recognize strangers. They'll come and talk right in your face, and they love everyone. They'll come up and tell you they love you, and dogs are kind of like that. Yes.
Bob, for your talk. I was actually just in Colorado this summer, and we didn't get to see the last two incredible, but they were saying that in like a month ago, that they were going to bring in 2 new packs. Were you involved, and did you get to tell them about those, what your findings on suggesting like what packs should be brought in there?
Yeah, that's a long story. We did consult with them, but then again, they didn't take any of our advice. So
That's a short story.
Okay, you're right. It went through many phone calls and everything else. Okay, thank you.